All right, so we're going to start talking about all of the membrane-enclosed organelles that are found in eukaryotic cells. And make sure you review which ones are unique to animal cells versus plant cells in the first video. So I've kind of clumped these organelles together into different groups based on what they do. So for example, the organelles involved in genetic control include the nucleus and ribosomes, which are what we're going to talk about first. So the nucleus is the most obvious structure under the microscope. When you look through the slides for the cells lab, you'll be able to see the nucleus easily under the microscope. So the nucleus is the structure that houses DNA in a eukaryotic cell. The DNA stays in the nucleus, and the DNA is read through the process of transcription to make the other nucleic acid RNA, which then has to leave the nucleus to get picked up in the cytoplasm and be read through translation to make a protein. So the DNA stays in the nucleus, and the RNA is made in the nucleus, and then the RNA has to get out of the nucleus. And the way that the RNA gets out is through these little holes called nuclear pores. So if we zoom in a little bit here, this is showing the phospholipid bilayer. The nuclear envelope, the plasma membrane-like structure that surrounds the nucleus, is not actually just a single phospholipid bilayer. It's two phospholipid bilayers thick. It's a double membrane. So the nuclear envelope is a double membrane. It's effectively four phospholipid layers thick. Okay, two phospholipid bilayers thick. And then the holes in the nuclear envelope are surrounded with proteins. And these holes in the nuclear envelope are called the nuclear pores. And the RNA travels through the nuclear pores to get out to the cytoplasm where it can be read. So the DNA stays in the nucleus. And another feature that you can see of the nucleus is there's this dark staining spot. That dark staining spot is called the nucleolus. Okay? So the nucleolus, what it translates to is little nucleus. And it's not a separate structure. There's no membrane or anything separating the nucleolus from the rest of the DNA in the cell. But instead, it's dark staining because lots of proteins that are involved in reading the DNA and making a special kind of RNA called ribosomal RNA are being synthesized here. So it's dark staining because there's lots of protein attached to the DNA there. So if we think about the process of reading the DNA, making RNA, and making proteins, most of the RNA that has the information for how to build a protein is called a messenger RNA. But the way that the messenger RNA, or mRNA, is read is with the help of a ribosome. And ribosomes are made of both protein and ribosomal RNA, or rRNA. So the RNA component of the ribosome is what's being synthesized in the nucleolus. Other vocab words that go along with the nucleus. The DNA that's in the nucleus is not just naked DNA. It's actually DNA wrapped around proteins. And that complex of DNA with the proteins that it's wrapped around is called chromatin. Another vocab word that goes along with this is the chromosomes. Chromosomes are linear DNA molecules, and in a cell that's not dividing, we actually can't see the individual chromosomes even though they're actually there. It's not until they are all the way condensed down and getting prepared for cell division that we can see these X-shaped structures of replicated chromosomes. So those are just a couple of vocab words that go along with the nucleus. So if we take that mRNA that's left the nuclear envelope through the nuclear pore and is now out in the cytoplasm, it's going to get picked up by the ribosome. The ribosome was our only example of an organelle that's found in both eukaryotes and prokaryotes. But ribosomes are not membrane-enclosed organelles. There's not a phospholipid bilayer that wraps around them. Ribosomes are made of protein that structurally holds everything together and rRNA, ribosomal RNA. And it's that rRNA that's the catalytic component. It's the enzyme that does the job of building proteins. So what the ribosome does, its function, is it reads the messenger mRNA, it reads the mRNA, and uses that information to build a polypeptide. In other words, what the ribosome does is catalyzes the formation of the peptide bond between amino acids. There's two different kinds of ribosomes, but they both do the same thing. Some ribosomes just float around in the cytoplasm, and they're called cytosolic ribosomes, and they'll make proteins that stay in the cytoplasm. Other ribosomes, on the other hand, are bound to membranes within the cell. So you'll see them on the nuclear envelope as well as the next organelle that we're going to talk about, which is the rough endoplasmic reticulum. These ribosomes are exactly identical as the cytoplasmic ribosomes. All ribosomes actually start out in the cytoplasm. But some proteins that are going to get put into our next organelle, the rough ER, or embedded into membranes, have a signal sequence that tells those ribosomes to go to the rough ER. And so the ribosomes on the rough ER will make proteins that either end up inside of the rough ER or stuck in the membranes of the rough ER. The rough ER is our first member of the endomembrane system. All that word means is the inside membrane. So the membrane-enclosed organelles inside of the cell that can pinch off little bubbles and fuse with each other and exchange their contents. So the endomembrane system includes the nuclear envelope, the endoplasmic reticulum, which can be both smooth or rough, the Golgi apparatus, lysosome, vacuoles, and these little vesicles, these transport molecules that move things from one part of the endomembrane system to the other, and eventually out to the plasma membrane. So the endomembrane system is in communication with each other. Little bubbles or vesicles can pinch off of one part of the endomembrane system and fuse with another part of the endomembrane system. And things that are being synthesized tend to go in the direction of the pink arrows, out, and some things can be brought into the cell and metabolized and brought back in. So there's a lot of movement within the endomembrane system. We're going to see for most proteins that go through the endomembrane system, they're going to start at the rough endoplasmic reticulum. They're going to get passed to the next member, which is called the Golgi apparatus, and then out to the outside of the cell. The endoplasmic reticulum is really made of two parts, the smooth endoplasmic reticulum and the rough endoplasmic reticulum. And they're connected to each other. So these inside components of the ER, the inside is called the lumen, is actually connected between the smooth and rough ER. But what distinguishes them is that the rough ER has these bumps on it, which are the ribosomes, where the smooth ER doesn't. Since the smooth ER lacks ribosomes, it has a different function than the rough ER. The smooth ER has three specific functions I want you guys to know about. First of all, it's a place where many lipids are synthesized. So we can add phospholipids to the endomembrane system here. Oils and steroids are synthesized in the smooth ER. A second function of the smooth ER is it contains enzymes for breaking down drugs, toxins, poisons, and metabolizing them. So in cases where you have someone who's habituated to drugs or alcohol, there's a neuronal response, but there's also a cell biology response. And in their liver, for example, the smooth ER is expanded so they can process a higher load of those particular toxins. Another function of the smooth ER is it stores calcium ions. And this is particularly important for our muscle cells. In our muscle cells, the smooth ER actually goes outside of the muscle cell fibers and wraps around it. And when a nerve impulse is received, the sarcoplasmic reticulum, which is the smooth ER outside of our muscle cells, releases calcium ions, which leads to a nerve impulse, and which responds to the nerve impulse by a muscle impulse, and the muscles contract. So the functions of the smooth ER are synthesis of lipids, breakdown of drugs, toxins, poisons, and storing calcium ions. The rough ER has a different function. The rough ER is the place where proteins that either will be built into membranes or secreted are formed. And I want to clarify this. The ribosome is the thing that builds the proteins. The ribosome is the thing that forms the peptide bond between amino acids. Ribosomes make the proteins. But the rough ER is the place. The rough ER is the location where proteins that are going to be secreted or built into membranes are built. Okay. So the rough ER is the location of secreted and membrane protein synthesis. A second thing that happens in the rough ER is those proteins that get pushed into the inside of the ER get sugar structures added to them. And so a protein that has sugars added to them is called a glycoprotein. And this process of adding sugars to proteins is called glycosylation. Once those proteins have been formed in the rough ER, little vesicles pinch off and are transported to the next member of the endomembrane system, which is called the Golgi apparatus. The side of the Golgi that's close to the ER is called the cis Golgi, and the side of the Golgi far away from the ER, closer to the plasma membrane, is called the trans Golgi. What happens in the Golgi apparatus is that those glycoproteins that were received from the ER get modified. So each time a vesicle will pinch off and fuse with the next layer of the Golgi, pinch off and fuse with the next layer of the Golgi, pinch off and fuse with the next layer of the Golgi. And in each of these layers are different enzymes that modify those sugar structures. And those modifications mark the proteins for their final destination in the cell. So this is the receiving side. The cis side is the receiving side. The proteins get modified as they go through the Golgi, and then on the trans side, they get transported to their final destination. Just a few more members of the endomembrane system. The next member is the lysosome. Now, recall the lysosome is one of those structures that are typically found in animal cells that are not found in plant cells. The function of the lysosome is to digest and break down macromolecules. So it can do this to structures that are brought in from the outside of the cell. So for example, if this were a white blood cell that phagocytosed or gobbled up a bacterial cell, that bacteria would be transported to the lysosome. And the lysosome is full of strong hydrolytic enzymes that will break down the bacteria, and then those parts can be recycled and used elsewhere in the cell. Cells can also break themselves down. The process of breaking down organelles that are no longer functional is called autophagy, self-eating. So the cell can build a new membrane around, say, a mitochondria that's leaking reactive oxygen species and transport it to the lysosome to break it down. A vacuole is a kind of catch-all term for other parts of the endomembrane system. And so there are different kinds of vacuoles in different cell types. So when that white blood cell brought in the bacteria by phagocytosis, that structure that wrapped around it was called a food vacuole. In organisms that live in fresh water, they can actually collect water into a vacuole and squeeze it back out, and that's called a contractile vacuole that helps balance the water regulation in those cells. In plant cells, they have a large central vacuole, which can be a storage organelle that holds pigments or dyes or oils, but it also takes up a bunch of space in the center of the cell. That allows plant cells to get bigger because they have this big metabolically dead spot in the center. So they still have a high surface area to volume ratio because this is an empty space in the middle of the cell. One last thing that I want you guys to think about is the fact that the inside of the endomembrane system is in communication with the outside of the cell. So if we have ribosomes out here in the cytoplasm, they're going to make proteins that end up trapped in the cytoplasm, and those proteins are going to remain in the cell. But when a ribosome sits down on the surface of the rough ER and pushes a protein either partway across the membrane or all the way into the lumen, and then little vesicles pinch off and go to the Golgi, through the layers of the Golgi into a vesicle, when that vesicle fuses with the plasma membrane, whatever was inside the vesicle is now going to be on the outside surface of the cell. So this little star-shaped part of the protein is going to face out towards the extracellular fluid, whereas this side of the protein is going to remain on the inside of the cell. So everything that's on the cytoplasmic side is going to stay in the cytoplasm. Anything that goes through the endomembrane system is going to end up on the outside surface of the cell.